Okay? So if I drew a recombination event in a single one, and I drew it randomly, I just happen to draw it right there. So what that would mean is that I have effectively swapped the bottom of this chromosome onto this side and vice versa. So I'm hoping what you notice is that following recombination, I won't have done anything to the relative positions of big R's and big Y's and little r's and little y's. Does everybody see why that would be the case? So if I then take this cell and I go to meiosis, and I go to metaphase of meiosis, and then anaphase, and separate the pairs of sister chromatids, what I'd end up with is I would pull this pair together to one side of the cell, and I would pull this pair together to the other side of the cell, and then I would split the cell. And so I'd end up with two cells that look like this. Now is meiosis over? We've only completed the first phase of meiosis. So we need to go into meiosis 2 and separate these again. So they would line up again, and then we would split them. We'd pull apart the sister chromatids from each other, as we've talked about, and we would divide the cell. And so you'd end up with two cells from this, and two cells from this cell. And this is what their chromosomal makeup would look like. So what would a Punnett square look like, again, if you were to have two individuals that are big R, big Y, little y, crossed together, where the genes are on the same chromosome, like they are here? What would the Punnett square for that look like? How would it be different from that crazy Punnett square you had to draw last time? So think about it. Try to sketch out a Punnett square for this situation.